This is Chaga fungus. And this Chaga fungus is quite uncommon. It's a parasitic fungus that almost exclusively infects birch trees. And estimates vary, but it's often found on only a small fraction of trees in any given area. And its growth is dependent on specific environmental conditions, a suitable wound on a birch tree, and of course the chance arrival of its spores. And so finding a Chaga conch on a birch tree feels like discovering a hidden forest secret or being in an adult treasure hunt. But I want to take a deep dive into this fungus. So today we're not just going to look at this rare organism, we're going to take it apart layer by layer. We're going to explore its unique life strategy as a sclerotia, and we're going to dive into the specific biochemical pathways it uses to create its protective armor. So first, let's be precise about what we're looking at. The structure you see here is not a mushroom. All mushrooms are fungi, but not all fungi are mushrooms. A mushroom is simply a fruiting body, a temporary reproductive organ designed to release spores. And this here is something else entirely, a sclerotia, as I said before. And a sclerotium is a compact, hardened mass of fungal mycelia. So, as I said, the Chaga fungus is parasitic on birch trees. It infects birch trees. And this does this, likely through a wound in the bark, and then it slowly spreads its mycelia through the heartwood of the tree. And to protect itself from the extreme cold of its habitat and to ensure its long-term survival, the fungus consolidates its energy and resources into this dense, woody conch structure. And it can remain dormant there and viable for decades, just patiently waiting for a favorable condition in order for it to reproduce. And that brings us to its most striking feature, the black, crusty exterior of the fungus. That is a highly specialized layer of protection, and its color comes from a pigment we all know very well, melanin. And it manufactures this melanin through a fascinating biochemical process. You see, many fungi synthesize melanin through at least one known biochemical pathway. That pathway is the 1,8-dihydroxynaphthalene pathway, or for short, the 1,8-DHN pathway. And that pathway begins with a simple precursor molecule, often acetyl-CoA, which fungal cells convert then into 1,8-DHN. And from there, a series of enzyme-driven reactions polymerize these molecules, linking them together into long, complex chains. And these polymers then become melanin pigments. And I'm of the opinion, and I'll justify this in a bit, but this isn't my area of expertise as a mycologist, but I believe, my hypothesis is that this 1,8-DHN pathway is the predominant melanin biosynthesis pathway used by the Chaga fungus. So this pathway is fundamentally different from how our bodies produce melanin. In humans, for instance, melanin is synthesized in specialized cells called melanocytes, primarily through the L-DOPA pathway. And the key enzyme in that pathway is tyrosinase, which acts on the amino acid tyrosine to produce the pigment that colors our skin and hair. Chaga, using this 1,8-DHN pathway, bypasses this L-DOPA route. However, that does not necessarily mean that Chaga can't use that L-DOPA pathway to produce melanin. To my knowledge, it is not definitively known whether Chaga can utilize both of these biosynthetic pathways. It may very well employ both pathways facultatively depending on its conditions. I don't know. Either way, this fungal melanin that Chaga produces is incredibly effective. It's embedded into the cell walls of the outermost layer of the sclerotia, forming this shield that is highly resistant to UV radiation, microbial attack, and physical damage. And some more interesting biological activity is also happening underneath that dark melanized armor. When we cut open a Chaga conch, if you will, we reveal this golden brown, corky, sterile tissue. And this is called the fertile contexture of the sclerotia. It's softer, it's more porous, and it's where the fungus stores its energy and produces some of the exotic compounds that Chaga is known for. And this inner tissue is in direct contact with the living wood of the birch tree, and it's here that we find the compounds that have drawn human interest for centuries. As the fungus slowly decomposes the wood of the birch tree, it extracts specific molecules from the birch and modifies them. And the most notable example is betulin. Birch trees are rich in betulin, which gives their bark its white color, among other things. But the Chaga fungus absorbs betulin and enzymatically converts it into betulinic acid. And other parasitic fungi, like the birch polypore, for instance, also enzymatically convert betulin to betulinic acid. But in Chaga, this conversion is a multi-step process driven by a sophisticated enzymatic pathway. The fungus synthesizes triterpenoids like betulinic acid using the mevalonate pathway, and this is the fundamental pathway for producing these triterpenes in fungi, where key enzymes like acetyl-CoA reductase and squalane synthase create this basic triterpene backbone that later forms these betulinic acids. And interestingly, the ability to modify this triterpene backbone into specific compounds is enhanced by significant evolutionary development. So genome analysis of Inonotus obliquus, the Chaga fungus, has revealed a major expansion in its cytochrome P450 gene families as a result of a whole genome duplication event that occurred approximately 1.3 million years ago. This genetic event provided the fungus with a much larger toolkit of specialized enzymes. Specifically, certain cytochrome P450 enzymes act on the betulin absorbed from the birch tree, modifying its structure to create betulinic acid and these unique triterpenoids. And this is a clear example of an evolutionary arms race. The birch tree first evolved high concentrations of betulin in its bark as a chemical defense against things like insect herbivory, and more importantly, for our purposes, fungal infection, and it evolved this millions of years ago. Betulin is toxic to fungi. It disrupts fungal cell wall synthesis and fungal cell wall membranes, halting their growth. The Chaga fungus later evolved this whole genome duplication as an expanded genetic toolkit to overcome that defense that birch trees evolved. So it turned the tree's protective compound into a resource for its own use. This whole genome duplication is an exceptionally rare event for a fungal organism, and this betulinic acid just so happens also to be a much more bioavailable form to human beings of betulin, which has its own beneficial, supposedly medicinal properties in human beings. But going back to the melanin pathway that I mentioned earlier, this whole genome duplication event that allowed Chaga to conquer betulin had a profound and unintended consequence. The same army of new cytochrome P450 enzymes that was used to process the tree's defense also provided the perfect toolkit for building its own melanin. The 1,8-DHN melanin pathway starts off with acetyl-CoA, which all organisms make in primary metabolism. So the first step is performed by a polyketide synthase. This enzyme acts as a molecular assembly line, taking acetyl-CoA and repeatedly adding other building blocks to it, and it then cyclizes and modifies this chain to create the first stable aromatic ring precursor, a molecule called THN. And again, that massive expansion of the P450 enzymatic family, driven by the whole genome duplication that Chaga experienced, gave Chaga the enzymes that perform the critical reduction and modification steps that convert that raw THN precursor into the final building block, 1,8-dihydroxynaphthalene, or, as I mentioned before, 1,8-DHN. And then the final most important step is the polymerization. The 1,8-DHN molecules are handed off to a lactase enzyme, and this enzyme uses oxygen from the air to grab the 1,8-DHN molecules and link them together into long complex chains. The result then is the black-brown eumelanin, the very substance that forms Chaga's famous protective melanated crust. The eumelanin that constitutes Chaga's resilient black armor is the same class of pigment responsible for the rich and varied dark skin tones celebrated in human diversity. In contrast, pheomelanin, the pigment that produces lighter shades of human skin and red hair, is not part of Chaga's biochemical repertoire, interestingly enough. But since I'm a plant pathologist and I like to think about these things, Chaga, of course, infects a very small percentage of wild birch trees, and certainly Chaga being parasitic off of them is not going to pose any existential threat to the species. But hypothetically, let's think of a scenario where Chaga is much more virulent and is threatening to cause the extinction of our beautiful birch tree. So as a plant pathologist, how then would I fight this? Well, I would first check out the RCSB protein data bank, which is the definitive digital archive of life's molecular machinery. It is the single most important repository for the three-dimensional structures of large biological molecules, primarily proteins. And this protein data bank, or PDB, holds experimentally determined 3D structures determined primarily using X-ray crystallography, NMR spectroscopy, and increasingly cryo-electron microscopy. And each file contains the precise 3D coordinates of a protein. So a protein's function is inextricably linked to its shape, and so then knowing the exact 3D structure of an enzyme, like a lactase or a reductase, allows scientists to understand how it works, how it binds to other molecules, and where its active sites are. And this then allows us to design drugs to modify its behavior. And so without the protein data bank, modern drug discovery, biochemistry, and biotechnology would just be impossible. And so then we use these structures to investigate and to design fungicides and herbicides as well. And so then I was just looking around out of curiosity. These are the things that mycologists do for fun on their off time. The structure of trihydroxynaphthalene reductase in complex with NADPH and an active site inhibitor. And this active site inhibitor is tricyclazole, which is a known fungicide. And the enzyme trihydroxynaphthalene reductase is one of the enzymes involved in this 1,8-DHN melanin biosynthesis pathway. And so I found this protein in the database, and here we can see the protein in this visualization software. Chimera X is the name of this visualization software if you're interested in downloading it. And this shows us how the tricyclazole binds to the active site of the trihydroxynaphthalene reductase, or 3-HNR for short, preventing the precursor molecule THN that I mentioned earlier from accessing the site, thus halting melanin biosynthesis. Now, the 3-HNR enzyme pictured here is not for chaga, that's an important detail. It is for the plant pathogenic fungi Magnaportha grisea, which is a plant pathogenic fungus which affects rice. However, chaga most likely also has a 3-HNR enzyme whose active site could be slightly different, but similar enough for this compound to cause fungicidal activity. And so using this class of fungicide on chaga could potentially halt melanin biosynthesis in chaga, which if we were trying in our hypothetical scenario to fight chaga's infection of birch trees, this would be a proper starting point. We would have to do X-ray crystallography to determine the structure of the chaga 3-HNR and determine if this tricyclazole would bind to its active site, and then prove it experimentally as well. But if we halt melanin biosynthesis, typically the chaga fungus needs that for its survival. How is the fungus going to protect itself from UV radiation if it doesn't have this melanin protective layer? And how is it going to halt fungal infections that the melanin layer usually protects from, and physical damage if it doesn't have the ability to synthesize melanin? These are the tricks that we use in mycology to fight plant pathogenic fungi. So that's all for today, folks. Thank you for joining me on this deeper look into the biology of chaga. And if you found this exploration interesting, please consider subscribing to the channel for more content that goes beyond the mycological surface. I'd love to hear your thoughts in the comments below, and until next time, stay curious.